in order to look at nervous tissues, we'll have to look at nervous system. And as you mentioned before, Daniel, tissues are a combination of cells organized together with specific function. We know that we're looking at different types of cells. They are highly organized, a bit more complicated in terms of arrangement, a bit more complicated in terms of architecture. So you won't expect the typical type of cell that we've looked at before, like the small cells, for example, in the connective tissue or in the epithelium lining cavities or body surfaces. You're going to expect cells that are very long, cells that have cell bodies or cytoplasm that is rich with different substances, and you're going to look at cells, they have external projection or cytoplasmic projection that are very long to basically carry out action potential. Similarly, we looked at muscle tissue. We know that skeletal muscle cells, the fibrils or myofibrils, they're very long. So these cells are very long, but when you look at cardiac cells, when you look at smooth muscle cells, when you look at epithelial cells, these are just cells that are typically flat or cuboidal, columnar, a bit more highly defined in shape. But when you look at nervous system or cells in the nervous system, they are highly complex. In order to look at nervous tissue, we'll have to look at nervous system. So nervous system is divided into central, peripheral, autonomic. Central nervous system, when we talk about central nervous system, we're talking about the brain stem, the spinal cord. So if you look at this image here, it's a simple diagram, but I would say take note of, for example, the brain, the cerebrum, and the different parts of the brain, frontal lobe, parietal lobe, occipital lobe. Look at the cerebellum. For central nervous system, we know that it includes the brain stem. Brain stem is right here, include midbrain, pons, medulla, the spinal cord, and this is the spinal cord. So all of that is central nervous system. Peripheral nervous system, you're looking at the nerve fibers like axon, dendrites, nerve endings. So if you look here, this is peripheral nervous system. Peripheral nervous system is divided in different structures based on where cells or neurons are emerging from different area of the spinal cord. Autonomic nervous system is peripheral nervous system, and autonomic nervous system can be further categorized or divided into sympathetic and parasympathetic. What this means is that the cells in autonomic nervous system communicate with cells in internal body cavities with organs. So you have these type of neuronal cells that are communicating with organs. For example, the cardiac muscle. You have cells from the sympathetic nervous system or neurons from the sympathetic nervous system communicating with the cardiac muscle, the smooth muscle of the stomach, which helps with peristalsis or contraction for peristalsis, also in the liver, as well as the bladder, and even other organs. So if you look at sympathetic nervous system is a branch of autonomic or peripheral nervous system. So these nerves or neurons, these cells are going to provide nerve supply to internal body organs. For parasympathetic nervous system, it does the same. What's the difference though? They have highly specific function. So if you look at the sympathetic nervous system, these nerve fibers actually come from different areas of the spinal cord, the vertebral column. You guys know the cervical, thoracic, lumbar, coccygeal region of the vertebral column.
I don't remember that exactly. I think it's 7 cervical.
7 cervical?
Yeah.
You sure?
No, I'm not sure.
How many thoracic?
It's at 13.
All right, so look, if you look at the image right here, you see T12, T1 to T12, right?
Oh, okay, yeah.
This is basically a better picture of what you should expect. Cervical region, as you said, how many nerves do you expect to emerge from cervical region?
8.
Okay, good observation. Good observation. All right, for example, thoracic.
Would that be 13?
Well, it would be 12.
Be 12,
How about
okay.
lumbar?
That would be 5.
Yes. The point is that these nerves leave specific region. For the sympathetic nervous system, these nerves leave the thoracic and lumbar region and supply smooth muscle in the gastrointestinal tract, also the cardiac muscle. And you also have parasympathetic that does the same. The parasympathetic, unlike the sympathetic, these nerves basically come from the cranial also cranial nerves. These nerves come from the brain, so you call them cranial nerve, and some come from the sacral region, so you call them craniosacral. So you have craniosacral nerves that basically comes off at different points from the brain, from the sacrum, and they basically go into supply, give parasympathetic supply to body organs. So basically, the sympathetic nervous system is like your flight and fright system. I'm sure you've heard that before, right?
Yes.
Yeah, so if somebody scare you, you get anxious, you start to run if you need to run in a dangerous situation. So you have the sympathetic nervous system that supplies specific organs in the body that are responsible to activate when there's a flight or fright response. And then there's a parasympathetic that opposes this. It relaxes you. It does somewhat the opposite of the sympathetic. So if you look at the sympathetic nerves that supply the eye, and it's far more complicated than this, I'm just simplifying it. The nerves that supply the eye, they are responsible for pupillary dilations. These neurons actually end on muscles like ciliary muscle and will basically cause the muscle to dilate. You guys looked at histology slides and you saw submandibular glands, sublingual parotid. You have also areas of the sympathetic nervous system that supplies these areas that will inhibit salivation, or for example, if it is a parasympathetic, it will basically stimulate salivation flow. The parasympathetic is the opposite of the sympathetic, where the sympathetic would dilate the pupil while the parasympathetic would constrict the pupil. And if you look at the heart, for example, if somebody scare you, you have a sympathetic nervous system activation where you produce certain types of neurotransmitter that will create an increase in the heart rate and increase in the blood pressure so that you can run. Whatever biological response you need in that moment in order to protect yourself from danger, that's basically what the body will do. And I guess we think we are intelligent by design because we are carrying out all of these actions, yes, but we have no control over them. It's autonomic nervous system. It's not like the somatic response or system where it's your voluntary action. For example, the skeletal muscle, pupillary dilation, salivation, racing of your heart, all of those are controlled automatically. In times of need, you will have gluconeogenesis or glycogenolysis. In this case, the sympathetic nervous system will cause glycogenolysis where you release glucose. The bladder, it does the opposite. In order to not go to the bathroom, it's a sympathetic response to have bladder contraction. You can learn this in your spare time. You don't have to feel burdened to learn it now, but in order for you to understand the basic things about even the cells that we're going to look at, you'll have to have a basic understanding as to this part. You already know most of these, the sympathetic versus parasympathetic nerve supplying to different organs. This shows the sympathetic fibers. The only difference is that these nerve fibers go to a ganglion. When I say pre-ganglionic and post-ganglionic, you'll understand that pre-ganglionic comes from the spinal cord, a specific area in the spinal cord, and it goes through a ganglion. For example, this is the sympathetic chain, and there are specific nerve fibers that goes to the specific ganglion, and then you have the post-ganglion. So the pre-ganglion is before the ganglia, as it makes sense, right? And then the post-ganglion is after. This is just showing that they all go through ganglion. For example, to go to the lacrimal gland, if you want to cry to produce tear or lacrimal fluid, you need the sympathetic versus parasympathetic activation of these glands. And these are those nerves that go to the glands. And it's just showing that they actually go through ganglion, and they have different names, ciliary, optic. You don't have to know all of that detail, just putting it into a bit more perspective. This is showing the same thing. It's basically just showing the ganglions, like ciliary ganglion, pterygopalatine ganglion, otic ganglion, submandibular ganglion. As I said, if we're looking at nervous tissue, you're not going to see cells that are as simple or easy to understand like epithelial muscle tissue. We think that we are intelligent, but we have almost no control over most of our actions, as you can see. These are just highly complex cells. Neurons are cells that are interacting with other cells. So it's either them interacting with a smooth muscle cell in the abdomen, it's with the cardiac myocytes or myofibrils or the skeletal cells. They're just interacting with them in order to create actions in them. So they're basically, I would say, like the god of the human body, right? These cells, because they're literally just directing all the actions. And from this perspective, you can see that we're basically a machine. Most of our actions are not so much within our control. This shows where we're actually in control, the somatic nervous system, the area in the brain where the motor response. Motor means like a muscular response. If you say upper motor neuron, lower motor neuron, it's talking about skeletal muscles. These are just nerves that have fibers that are going to the skeletal muscles to create voluntary action. There are different areas in the brain where you have motor neurons. So if you look here, we look at the frontal lobe, mental note of the frontal lobe. You have specific area in the brain where these nerve cells come from, go down through the spinal cord. They go through specific areas in the spinal cord, then have contact through the spinal cord to the muscle itself. So you will see that there are muscles, skeletal muscles, and there are nerves running on those muscles. These nerves are connecting with other components in the peripheral and the central nervous system in order to create that voluntary action. So that's the somatic nervous system. The somatic is different from the autonomic in that, what would you think the difference between the somatic and the autonomic nervous system would be in that case? It's very simple. If you understood anything that I just rambled a while ago.
Somatic nervous system basically involves voluntary control, whereas the autonomic nervous system involves involuntary control within the body.
Exactly. Perfect. See that you're so sharp, Daniel. You can give me an example of what an autonomic control would be? An example of sympathetic versus parasympathetic response?
I would say peristalsis. Maybe that's something,
Mm-hmm.
As food moves through the digestive tract, that would be
Perfect. In order to look at nervous tissue, we'll also have to look at the embryology of nervous tissue. So nervous tissue derived from ectoderm, and you'll get a little bit more detailed embryology in this one, because this one, I think we need to understand a little bit more from this level. We know it derived from ectoderm. A basic thing that we understand, there is a neural plate that appears at around 20 days. 20 days, you have a lot more cells. 20 days, remember you start from the zygote, the morula, the blastocyst, the different cell stage. At 20 days now you'll have a neural plate. This is the neural plate here. And what will happen is that within the midline of the neural plate, you will have a neural groove, and it goes all the way down. And if you see, can you think of anything that in your body that goes all the way down like this? Think about it. Spinal cord, right? All the way down to the middle, the neural groove. But this will basically form the spinal canal. This is like an opening where there is this groove. So this neural groove will be the central canal in the spinal cord. So basically it holds, and this will form a tube. Just think about it like a plate, like a plastic plate that cannot break, and you fold it on the side, and you create a tube out of it. So that's called a neural tube. This neural tube will form the brain and the spinal cord. So this entire fold, brain and spinal cord, and this groove will be the central canal, and you'll see what the central canal looks like. If you look at a histology slide of the spinal cord, you will see the central canal. In that sense, you can relate the embryologic origin to a neural groove, right? And after the neural groove, if you look, there are some cells that are called neural crest cells. You'll see that the neural crest cells give rise to a lot of cells. So the neural tube and the neural crest cell give rise to a lot of cells. The neural tube give rise to also the ventricles. Anybody know what the function of the ventricles are? What would you have in the ventricles? of the brain?
Would they be involved in the limbic system?
Not entirely. It's a fluid-filled ventricles. What kind of fluid do you think would be in it? You know of any fluid in the brain?
I know fluid. I don't know the
Cerebrospinal fluid, right?
Yeah.
You have to know that there is cerebrospinal fluid. Your body produces around 500 mils per day. These ventricles are what will contain different cells that will make the cerebrospinal fluid. This is just looking at the lateral ventricle. There's an anterior horn of the both ventricle, posterior horn of both ventricles. Lateral ventricles drain into the third ventricle that drain into the aqueduct of Sylvius and into the fourth ventricle down through the spinal canal. You'll realize that there are specialized cells that are responsible, similar to the epithelial cells, but they're not the epithelial cells. Look, they are called ependymal cells. These are specific cells that are in the ventricle. This image is showing the ventricle here. There's a thing called choroid plexus in the lateral ventricle, and they actually have cells, ependymal cells. Similar to epithelial cells, they have also tight junctions on either side, and they are responsible for producing cerebrospinal fluid. And if you don't understand the rest of the diagram, it's basically just showing the flow of cerebrospinal fluid and how the cerebrospinal fluid is filtered through here, an area called arachnoid granulation. And you'll understand a bit more about the dura pia and arachnoid mater. These are just coverings over the brain that are just there to protect the brain. They have different layers or different coverings, and you'll eventually know that they're just connective tissue. So once you understand so many things at the cellular level, you understand a lot of things in medicine. These neural crest cells and neural tube will give rise to all the cells that you will look at in the central nervous system. So the cells migrate on either side and form somites. So if you look, we were looking at different areas of the spinal cord or the vertebral column, cervical, thoracic, lumbar, all of these regions. They're just basically showing that the embryologic origin of them. So they form, the neural tube forms somites, and they eventually give rise to the cervical nerves. This is just showing everything, a compilation of everything that I just said. At 21 weeks, you have the neural plate. The neural plate is folded. When it's folded, you call it a neural fold. And after it's folded, you have a neural groove, which we said was the central canal. And then you have this neural crest cells that are also derived here. Neural crest cells that give rise to most of the cells. So the neural crest cell give rise to the cells in the peripheral nervous system, and most of the neural tube cells give rise to the cells in the central nervous system. This is just showing axon and dendrite. You'll get to understand that there are two types of cell in the central and the peripheral nervous system, just two. It's so complex, but they're just two types. You call them neurons or neuronal cells and glial cells. There are different types of glial cells, but once you know that there are just two types of cells, and one are the neurons, you'll eventually understand that basically is a long cell or a nerve cell that contain a cell body, just like you have your cytoplasm. You have the cell body soma with a nucleus. The difference is that you have a thing called axon, that's a long process that extends from the cytoplasm of the cell, and these axons are there to propagate action potential. You also have dendrites, and dendrites are there to also act as receptors or sensory receptors, motor receptors. So they are just long extension on these cells that are contacting muscles, contacting different cells in the body to create an action. This is just showing different areas of the brain are developed in embryology. You have a forebrain, midbrain, hindbrain. You can do this in your spare time. Know that the forebrain, hindbrain, eventually you'll end up with the cerebrum, the thalamus, the cerebrum that we looked at, thalamus, epithalamus. There's also the cerebellum. There are just different areas, pathway and division during the embryologic period that give rise to different areas of the brain. Thalamus and the hypothalamus would derive from diencephalon during a stage in the embryologic period. Mesencephalon would give rise to the midbrain. This is just showing how disgusting you look at some point during during your life before you knew that you exist, and the different areas of the brain that develop. For example, it's showing the mesencephalon, the diencephalon, but eventually the end result is that you end up with cerebral hemisphere, the two big hemisphere that you have. You end up with something called thalami, midbrain, cerebellum, and the ventricle. So this is the important diagram that you will probably need to pay a little bit more attention to. The neural crest cells give rise to different types of cells. The neural tube give rise to different types of cells. So if you look at the cells that are derived from the neural tube, you will understand that most of the cells in the central nervous system derive from the neural tube. You will also know that most of the cells from the peripheral nervous system arrive from the neural crest. So these are just showing the same thing. You have axons, but once you understand that there are different cells that derive from neural tube, and these include ependymal, oligodendral cells. There are also neurons. So once you see axons, dendrites, they're just neurons. This diagram is showing a lot of different cells. They're all just two types of cells on it. These are the neuronal cells, axon, these type of cells with cell body axon dendrites, neuronal cells. These are oligodendrocytes, ependymal cells, they're glial cells. These are also in the peripheral nervous system. There are the neuronal cells, and there are also the glial cells. The glial cells are going to be different though in the peripheral nervous system. You'll have cells called satellite cells, Schwann cells, and these cells have different function. This is just putting a little bit of clinical correlation to it. You already know that when this neural plate folds, it forms the groove. When it folds, it should fuse at the front. Sometimes there is no fusion, and as a result, depending on the area where it doesn't fuse, you have a baby that's born without a brain. You have the forebrain that would be deriving from this ventral, anterior ventral area. From this head portion of the neural tube, you should have the brain. If this doesn't fuse properly, then the brain won't form. This is called an encephaly or an encephaly, which means that the baby is born without a brain. And then if it happens at the back, you will also have a defect. It can be spina bifida, can be myelocele, meningomyelocele, which means that there's a defect in the posterior wall because the posterior pore, it did not close. So you basically can have the meninges, spinal nerve could be sticking out of it as a result of failure of it to close. There are different layers that cover the brain, as I mentioned before, the dura, pia, and arachnoid mater. These are just connective tissue, and they are situated at different areas in the brain. The dura is in contact with the skull. Below the dura mater, you will have a space. The arachnoid mater is also connective tissue, and you'll eventually understand there's the innermost layer, the pia mater. This is in direct contact with the brain. So the pia mater is in direct contact with the brain. The arachnoid mater is above the pia mater, and you also have a space between here. This space between here is basically where the cerebrospinal fluid flows, and you call it the subarachnoid space. So it's between the pia and the arachnoid mater. The cerebrospinal fluid is made in the lateral ventricle in different areas, and they're produced by small cells called ependymal cells. We'll actually look at the ependymal cells eventually. This is just showing different layers of the brain, giving different perspective again, that you have the outermost dura mater, the arachnoid mater, and there's also the innermost pia mater. And between them, you have different space. Sometimes you can have bleeding between the spaces, and depending on which area you have bleeding, give it a different description. For example, in old patients, sometimes they can have bleeding in the subarachnoid space because the veins are thin. So as you get old, the veins are thin in this space, and as a result, you can have bleeding between the dura and the arachnoid mater. So you can have subdural hemorrhage. The dura is the outermost layer, it's thickest, and it's dense fibrous connective tissue with flattened fibroblasts. There's collagen and elastic fibers. And we're talking about the spaces as well. This is showing the different space. So this is the pia mater, as we said, the innermost layer that is in contact with the brain. And there's the arachnoid membrane. It's all connective tissue, right? And there's a subarachnoid space. And remember, we said that the subarachnoid space is where the cerebrospinal fluid is circulating the brain. brain and in the spinal cord and around the spinal cord itself. So these are, we know they're meninges, dura, arachnoid, pia, in that order, or pia, arachnoid, dura. This is just showing histology again. The dura mater, it's connective tissue. Dura mater here, arachnoid, pia. They're just 3 layers. So you can imagine that your skull is here, and you have the dura mater. It's just a connective tissue here, and then you have the arachnoid below the dura, and then you have the pia below the arachnoid. It's just that you have spaces in between, and this space is basically the subarachnoid space where the cerebrospinal fluid will circulate. This is just showing the flow of cerebrospinal fluid, as we said before, made in the choroid plexus here in lateral ventricle, and they're just circulating between in the subarachnoid space. Sometimes you can have bleeding or rupturing of vessels in the subarachnoid space as well, and you have to do what is called a lumbar tap for diagnostic purposes, and then you'll realize that the cerebrospinal fluid that is supposed to be very clear is red or have a different consistency. If you have an infection, sometimes you'll be surprised what comes out of the subarachnoid space and how the cerebrospinal fluid is affected. In terms of clinical correlation, you can have inflammation of the meninges. Remember, the meninges are the dura, arachnoid, pia, mater. These are connective tissues that are covering the brain. You can have inflammation of the brain or inflammation of these tissues or connective tissues. So this is when viruses, bacteria invade these fibrous tissues and cause inflammation. So in terms of the central nervous system or the brain itself, the brain have different areas, gray matter, white matter. The arrangement of the gray and white matter is different or varies whether you're in the brain or the spinal cord. Gray matter is gray because it's just the cell bodies, and you look at the cells, eventually you look at the cell bodies and realize that there are substances in the cell that are creating this gray structure. Nissel bodies, or it's just the cell bodies. There are substances in the cell body, well, not substance, but structures called Nissel bodies. And if you would take a wild guess as to what the Nissel bodies are, they're rough endoplasmic reticulum. So they're just rough endoplasmic reticulum being visible because they're so dense in the cell body. So they appear as gray, or they call them gray matter. The cerebrum, this might look a bit complicated, and it's complicated, yes, but it's also very simple. So simplicity is good sometimes, but there is also element of complication. As long as you have a method to the madness, I think that's the idea. So there are neuronal cells, nerve cells, and glial cells. That's all that's here, 2 types of cells. The important thing though is that different areas look differently because you're seeing different areas of the cells. Here, you're seeing a lot of cell bodies. You're seeing a lot of cell bodies. Go to another areas, you're seeing a lot of axons. Cerebrum, this is the gray matter here, and you know why it's gray. You remember why it's gray?
Because of the cell bodies being located there, and you have the rough endoplasmic reticulum that stains very deeply.
Because what? Because of
you said the rough endoplasmic reticulum, because they're the cell bodies located.
Yes, exactly. So you call them Nissel substance, right? Or Nissel bodies. So it's rough endoplasmic reticulum. Very good. You have a keen ears for detail. That is very good because it's easier to understand. When you can connect the small things, it's easier to have an understanding of more complex things. So yeah, there are just nerve cells and glial cells that are here. So you have the gray matter and the white matter, different areas. Here you have neuronal cells and glial cells. So it's just a bunch of neuronal cells and the glial cells. And the neuronal cells, cell bodies will have Nissel substance, rough endoplasmic reticulum. White areas or different areas might just be the cytoplasmic projection, the axon that is myelinated, that's just running through tracts or different areas, trying to communicate with areas in the spinal cord, trying to communicate with areas in your smooth muscle, cardiac muscle, skeletal muscle. So these are just cells having different arrangement trying to communicate. This is the spinal cord. The spinal cord, the gray area is a bit differently arranged. Cerebrum, the gray area was on the external surface. If you look at the diagram closely, this says the pia mater. You know what's above the pia mater by now, right? The arachnoid and the dura, and after after the dura, you have the skull. You know, the pia is the outermost layer of the brain. Then you have the gray matter in the outermost layer of the brain, the white matter on the inside. If you look at the spinal cord, it's a bit differently arranged. You have the gray matter in the spinal cord that is located on the inside, not the outside this time. The gray matters are on the inside and the white matter on the outside. And you have an idea as to what the gray and white matter is, right? Okay, so yeah, this is just showing different neurons. For example, motor neurons. Remember, we say anytime we mention motor, it's going or carrying signal or nerve fibers for voluntary action. So these are just motor neurons, nerve cell bodies that are sending axons, sending projections to the muscle itself. And they're just located at different areas, very highly specific areas in the spinal cord. This is also showing a stain of the spinal cord. Remember, we said that you have the central canal. Central canal, we touched a bit on the embryology, the neural groove gives rise to the central canal. And you have the cerebrospinal fluid flowing in the central canal. Remember, it's coming all the way from the brain, from lateral ventricles to the third ventricle to the fourth ventricles down through the central canal. So it's just the cerebrospinal fluid. I guess the idea is for cushioning. This is the ventral. Ventral is usually anterior or the front. If you look at the ventral area, this is where the motor neuron running. The nerves are neurons carrying action potential for muscles. They're running through this area, the anterior ventral area of the spinal cord. Neuronal cells and supporting cells. And we said that the spinal cord, two types of supporting cells are the Schwann cell that is responsible for myelinating the axon in the peripheral nervous system. And there are also the satellite cells. But when you look at the central nervous system, you have different types of cells. We talk about oligodendrocyte. If you remember that diagram that I was showing you, oligodendrocytes talk about microglial cells and also ependymal cell for central nervous system. This is looking at the cerebellum, showing the arrangement of white matter as well as gray matter. The white matter is a bit more on the external region here. And there are different layers, a molecular layer, Purkinje cell layer because of the different types of cells that are there, and the inner granular layer based on what it looks like. These look like granule, and these are the Purkinje cells. So if you look at the outermost layer, they're unmyelinated neurons, and then you have Purkinje cells that are here and the granular layer that is here. So if you look at the histology of the cerebral cortex, you're supposed to see it somewhat if you have a good stain. You're supposed to see the cerebellar cortex and different layers. Two major cells in the CNS and the PNS, you already know by now. The nerve cells are neurons, the glial cells are supporting cells. This is the nerve cells up here, and these are the neuroglial or glial cells. Two types of cells. The nerve cell is highly complex. It's different. Have you seen any cell like the neurons? No cells like the neurons. No cells with these long projection connecting to other cells. The neurons or nerve cells are very specific, highly organized in architecture. We already talked about the cell body. We talk about the Nissl body or rough endoplasmic reticulum. Like any cell, you should expect the Golgi, the mitochondria, especially these cells, because these cells are highly organized, they're highly intelligent for highly specific function. So we should expect that these organelles that we've covered in the first session, because we're still talking about cells, we're not going to talk about anything different. We're going to be talking about nucleus, endoplasmic reticulum, mitochondria, Golgi complex, and the function of these cells the same. Mitochondria for energy, ATP for energy production. Golgi, you need it for packaging. The endoplasmic, rough and smooth, you need even with the ribosome for protein synthesis. So you're going to expect that a lot of these things will take place in neurons. And there are also the external projection of the neuron. You have microtubules or even protein filaments that are just there to transport things here from the cell body to this end of the cell. So this is the neuron or the nerve cell. This is one type of cell that is found in the central and peripheral nervous system, and it's the most abundant. And then you have these type of cells, which we call glial cells. These cells are just there to support the neuron. Really and truly, the neurons are the master of the household, basically. The neuroglial cells are there to support the neurons. There are different types of neuroglial cells, though, in the central nervous system versus the peripheral nervous system. And if you listen keenly, you would remember that I said the peripheral nervous system contains two types of glial cells. We call them satellite Schwann cells. This is easy to remember because the two of them start with S. Central nervous system now contains ependymal cells. We touched on the ependymal cells before. We said that they are in the ventricle to produce cerebrospinal fluid. In the central nervous system, it's important to emphasize that the oligodendrocytes are there to myelinate these axons. So you see these axons up here that are extending from the neurons. These oligodendrocytes are going to be wrapping around different segments of them, creating what we call a myelin sheath. It's like a lipid structure around it. And these myelin sheets that are being created by the oligodendrocyte in the central nervous system are there to help to propagate rapid action potential. The astrocytes, we spoke about blood-brain barrier, blood-testis barrier, blood-ocular barrier. We were talking about tight junctions. Anybody remember that? So the astrocytes are those cells that are there to help to seal the tight junction or create the tight junction as well. What would be their primary function in the brain?
To regulate the substances that can enter and come out of the brain.
So the BBB, the microglial cells, we touched on a type of cell that is phagocytic. Anybody remember? It starts with M2.
Macrophages.
Macrophage. So the microglial cells equivalent to a macrophage, where it's a phagocytic cells in the central nervous system. It's a phagocytic cell in the central nervous system. In the peripheral nervous system, you have Schwann cell. They are similar to the oligodendrocytes, where they are responsible for myelinating the nerves in the peripheral nervous system. You have a question? Anybody? No? Okay. So the neuron, we already touched on the two types of cells, the neurons and the glial cells. The neuron, you're just looking a bit up close at the neuron. It consists of a cell body or a soma. And there's the dendrites that we said that is projecting on one end, and there's also the axon that is a single process that is carrying impulses away. So the difference is that the dendrites, look at this end of the cell, this is the neuron. You have the dendrite. The dendrites are projection from the cell body, and the dendrites are responsible for taking impulse towards the cell body. So the dendrites will carry signals to the cell body, but the information will travel along the axon to the cell bodies and will either carry the information from the axon to the cell body back to the dendrites, or the other ways. The information can be both ways. You call it anterograde or retrograde, so it can go forward or backward. So this is just showing the what would you assume this would be? We talked about this before, and
Nicer.
Exactly, the Nissl substance, the Nissl substance being stained here. So the neurons can be classified into multipolar. Multipolar means that it has one axon and several dendrites, or it can be bipolar, where it has one axon, one dendrite. The image is showing here the one dendrite. These are just dendritic spines, even though it looks like a lot of different dendrites. So it's one dendrite, there's a cell body, the axon. So one axon, cell body, dendrite. Unipolar, one axon, unipole. So the cell body, axon. Cell body is that they have the cell body, axon, or you have the cell body, axon and dendrites, cell body dendrites. So it can be multipolar, meaning that one axon but several dendrites. Bipolar, one axon, one dendrite. Pseudo-unipolar, where it's just a short process that's just extended into an axon and a dendrite. This is just showing also the same thing that I mentioned before. It can be multi-uni, pseudo-unipolar, depending on the amount of axon and dendrites that are projecting from the cytoplasm. So this is an up-close look of the soma. We already spoke about dendrites before being the external projection. You also can have dendritic spine. And this is a closer look at the rough endoplasmic reticulum. As we mentioned before, these are Nissl substance. We call them Nissl substance in the brain. You just have a lot of rough endoplasmic reticulum. They're a Nissl substance in the brain. You expect the nucleus, you expect a lot of mitochondria, and you expect your Golgi apparatus, the lysosomes, and this is the axon. The initial segment of the axon is basically where you have action potential being generated most often. And then you'll realize that the cells, there are cells that are going to contact different areas of the axons, where we said that there were oligodendrocytes in the central nervous system. They will contact different segments of the axon to myelinate it. There would also be the Schwann cell in the peripheral nervous system that will connect a segment on the axon to myelinate it, and contacting that area or the areas of the axon, wrapping the membrane around the axon will help to insulate it and create or help to propagate rapid action potential. There are different areas without myelin sheath, so that's a big difference. So the soma also has microtubule. I think I mentioned that the microtubules, they're just long projections that are extending into the axons that are transporting neurotransmitter vesicles, all of these things. You'll eventually understand that in order to create an action potential, a lot of things has to be happening. A lot of things will have to happen in this cell or this neuronal cell, nerve cell, in order to create action potential. So there are going to have to be neurotransmitters involved. These are chemical proteins, amino acids that are being produced within the cell, transported to the terminal of the cell in order to release. When it releases these neurotransmitter at the nerve terminal, that's when we start talking about presynaptic, postsynaptic. Neurotransmitter can be released or at the presynaptic terminal, and it goes across an area where we call it synaptic cleft, where it communicates with the other cell, and when it contacts the other cell, that's when you call it postsynaptic terminal. So it's like information traveling from one cell to the next, and we talked about gap junction before. So that's the bigger mechanism. This is just an image of microtubules and neurofilaments. The neurofilaments, they are important for also regeneration of neuronal structures after injury. This is showing action potential muscle tissue. This is the nerve contacting the muscle. And we spoke about neuromuscular junction before, nerve-muscle junction. This is the nerve, as you can see. If you look closely on the diagram, it looks big and wordy. It's nothing that is too complicated. If you look closely, the neurolema is basically just the membrane or the cell membrane of the nerve. This is the myelin sheath. And if this is the myelin sheath and this is touching a muscle, where exactly would you think this is? Central or peripheral nervous system?
Peripheral
nervous system. So the cell that you would expect to be myelinating this axon via Schwann cell, right?
Yes, miss.
Schwann cell. This is the Schwann cell myelinating the axon in the peripheral nervous system. Schwann cell, they have them nucleus, they have the mitochondria, everything that's necessary to assist to aid in the action potential. But here you're looking at the nerve terminal where the synapse occur. We talk about release of neurotransmitter. Neurotransmitter, as we mentioned before, will be transported down to the axon terminal, and they can be released in vesicles across the neuromuscular junction. So you would have, for example, acetylcholine as a neurotransmitter being released from the nerve terminal across the synaptic cleft to the neuromuscular junction, and this will cause a binding of acetylcholine receptor on the cell. So when the acetylcholine receptor is stimulated, what it will do, it will create an influx of sodium into the muscle cell. And remember that when the sodium goes in, what will it do? It will trigger an action potential. So the calcium ion will be released from the sarcoplasmic reticulum. And you remember the sarcoplasmic reticulum is equivalent to the endoplasmic reticulum. So that's basically an action potential in a nutshell, coming from the brain to the axon, through the cell body, wherever that is, to the nerve terminal, or to the neuromuscular junction. If you remember anything about architecture, look at these dark, light, dark. You know that this is the sarcomere. These are likely to be mitochondria along sarcomere. Remember that we need them for ATP, for contraction. So actin, myosin filaments, these are what you're looking at, actin, myosin filament, nothing that you've never seen before. So the neurotransmitters can be different types, and I don't think I've mentioned that neurotransmitters are just chemical or protein. Yes, I think I mentioned it. Protein amino acids that are released at nerve terminals that will create a communication between another cell. It can be a nerve cell, or it can be a muscle cell, a smooth muscle cell, whatever it is. These things need to be released at the end of the nerve, or the terminal area of the nerve, or the axon, to basically stimulate the other cells. So the neurotransmitters can be glutamine, epinephrine, norepinephrine, serotonin. You don't have to know them, but you already understand the sympathetic nervous system. For example, the sympathetic nervous system will trigger a release of norepinephrine, and this will stimulate or activate the flight-fright mode. So the heart rate will increase, the blood pressure will increase, and you will respond. This is just showing the different neurotransmitters and how they interact at the nerve terminal and how they interact with receptors. Glutamine is a common neurotransmitter, acetylcholine, norepinephrine, common neurotransmitter. And these neurotransmitters are just released at the terminal. Sometimes they're released in vesicles, sometimes they're just released to interact with receptor on the other cell. When they interact with the receptor on the other cell or the terminal cell, it creates an action potential or stimulate the cell to do something.
Excuse me, miss.
Yeah.
Is it the glutamate receptor that alcohol interferes with?
Glutamate?
Yes.
Glutamine receptor?
Yeah, the receptor that the glutamate binds to. Is that what alcohol interferes with?
Yes, I would say a vague memory, yes. I think I deleted a slide that was looking at the clinical correlation, and I think alcohol would interact with the glutamine receptor. But I have to double check, not from my direct memory. But I think I deleted a slide that was looking at clinical correlation where that was concerned. I think I deleted a lot of slides because I have a problem with information or hoarding information. But yeah, thanks for bringing that up, Daniel. You give me more things to pay attention to. Okay, so synapses can either be excitatory, inhibitory. You don't have to know anything about the detail of that. It makes sense. One can create a stimulation for excitement. One can create an inhibition. Just like you have the sympathetic, parasympathetic nervous system. One will, for example, stimulate pupillary constriction. One will stimulate dilation. One will stimulate the heart to beat fast. One will stimulate the heart to beat slow. Either way, most of the organ systems that are controlled by autonomic nervous system are controlled automatically. There's an action and there's also an opposite to the action. So this is just the excitatory, inhibitory synapse. So in terms of synapses, there is the presynaptic terminal. When a typical synapse is carried out, there's a presynaptic terminal. There's also the synaptic cleft, postsynaptic membrane. This is just showing areas where the neurotransmitter is released. For example, if it is released from this cell would be presynaptic neuron, and the synaptic cleft is just the area between where it communicates with the other cell. So sometimes, as I said, it can be released in vesicles, can be released where it binds to the receptor on the postsynaptic membrane or the cell itself, and this will create or stimulate that cell to do something. So for example, if it was the neuromuscular junction, it would stimulate muscle contraction in a more complicated way, obviously. This is just saying that neurotransmitter is released by exocytosis, something that we've covered before. And just for repetition, there are two types of cells in the nervous tissues, two types of cells. So if I ask you that as a question, can't get it wrong. Neuronal cells, glial cells. And this is just a picture of what the neuronal cells look like. What's in the neuronal cells, it's no different from any typical cells that we've looked at before. It's different, yes, in architecture, in arrangement, and in terms of cytoplasmic extension. It's a bit different, but it's more highly organized, however, with more complex activities or complex actions to be facilitated. This is showing two types of cell in the nervous system again. This is showing the neuronal cells, as you already know. This is just showing the different types of cells in the central nervous system, and these are glial cells. So the glial cells, we already touched on the ependymal cells. The ependymal cells are similar to epithelial cells. They have cilia on the top, or microvilli. Obviously not the stratified squamous epithelium, but in terms of the arrangement, they're stacked, they're flat, and they have between them tight junction. They have cilia or microvilli on the top or apical surface. You already know about apical projection. There's also the microglial cell in the central nervous system. I remember that the microglial cells are phagocytic cells. There's also the oligodendrocytes, and remember that the oligodendrocytes are there to myelinate the axons, and as you can see, it's myelinating an axon. But what this is also showing is that the oligodendrocytes can myelinate more than one axon. One cell can myelinate more than one segment of the axon. One cell can myelinate two different nerve cells or neuronal cells. And as you can see, this one oligodendrocyte is myelinating this area or this segment of the axon, and it's also myelinating this segment of the axon. And you'll realize that this is only in the central nervous system. The one cell in the peripheral nervous system are only able to myelinate one segment of one axon, not one segment of more than one axons, basically. Then you have the astrocytes, which we already touched on, that helps to form the blood-brain barrier. You'll eventually understand that the astrocytes are food processes, that they extend on the endothelial cells and over the endothelial cells where there are the tight junctions, and that's how they create the blood-brain barrier. So they prevent drugs, chemical, different macromolecules from entering the brain. So the four types of glial cells in the central nervous system, you definitely need to know this. You already know that there are neuronal cells and glial cells. You definitely need to know that there are just four types of glial cells in the central nervous system. The astrocytes, you already know about astrocytes, the oligodendrocytes, the ependymal cells, microglial cells.
Excuse me.
Yeah?
Oh, all right. It looks like you went to speak about them now. I was going to ask about the astrocytes. I was going to ask that, so they not only form a barrier around the capillaries, but they also form a barrier around the individual nerves.
What, the astrocytes? Yes, they also form a barrier around areas of the nerves as well. They have a lot of functions. It's not only for the blood-brain barrier. They're important for growth. They're important for signaling function. They're important for many different things. You have different types of astrocytes that have different function, and they have different names in different areas of the body. For example, there was a diagram with the retina. The Muller cells in the retina would be like the astrocytes. Remember, we talk about the blood-brain, the blood ocular, and we're looking at the retinal pigment layer with different types of cells. The Muller cells in this case would be similar to the astrocytes. And we already touched on the fact that they form tight junctions on endothelial cells. But as you mentioned, Danielle, they also form tight junction on the neuronal cells as well. So in terms of the clinical point, astrocytes can create tumor. One thing you should know about tumors in the brain or central nervous system or peripheral nervous system, they usually only arrive from the glial cells, not from the neuronal cells. So the glial cells, like ependymal cells, you can have an ependymoma. From the astrocyte, you can have an astrocytoma, for example. And even in the central nervous system, where the oligodendrocytes can also be affected, where they are unable to myelinate a cell, or there is demyelination as a result of the oligodendrocyte being damaged or being ineffectively produced. So as a result, you have certain clinical conditions that can result as a result of not having the oligodendrocyte myelinating these axons. For example, in multiple sclerosis. A bit more on the blood-brain barrier. The blood-brain barrier is basically to restrict the inflow of macromolecules, as we mentioned before, and it does this with tight junction. There are tight junctions between the endothelial cells. There are also tight junctions between the foot process, being anchored between the foot processes of the astrocytes. The foot processes cover more than 85% of the endothelial cells. So these foot processes that extend from the astrocytes are pretty large. They cover up to 85% of the endothelial cells. So areas in the brain where you don't really have tight junctions, like where you have the glands, like the pituitary pineal, you don't really need the tight junctions so much because you need easy diffusion of these molecules. For example, when they're produced, hormones, when they're produced, you want them to be diffused easily, so you wouldn't have blood-brain barrier in those areas. But the tight junction allow for potassium and neurotransmitter to pass through it, however. So in terms of the oligodendrocytes, we already mentioned that they produce myelin sheath in the central nervous system. These are just images showing the oligodendrocyte. And in terms of the arrangement of the myelin, it's just overlapping of the cell membrane, the lipid membrane portion of the cell. It wraps around it and form multiple layers. So we already mentioned that the oligodendrocytes myelinate more than one areas or segment of different axon. One oligodendrocyte can myelinate different nerves. One cell in the peripheral nervous system can only myelinate one axon. This is just showing up close what myelination looks like. It's showing the fused layer of the cell membrane. So the oligodendrocytes, there are different layers, or the membrane of the oligodendrocyte, the cell membrane, overlaps repeatedly and they fuse. And because this is a lipoprotein structure, it creates a thick protein structure over the axon to prevent anything from passing through it, so that when there is action potential, fat is a good insulator, lipid is a good insulator of electrical conduction. It moves rapidly through the axon as a result of the myelin sheath. Between these myelin sheaths, you have spaces, and these spaces, you call them nodes of Ranvier. So remember that the cells are myelinating segments, right? So this is one segment, one segment, one segment of the axon. The area here would be where you have the nodes of Ranvier. So the nodes of Ranvier is between the segments, and these nodes of Ranvier contain calcium channel, and these channels also help to reinitiate the action potential. So between when the action potential, for example, is being propagated from here to here to here, there is the ion channel that has to be activated within the nodes of Ranvier that will reinitiate the action potential at each segment. So we touched a bit on multiple sclerosis, where you have loss of the oligodendrocytes. If you have loss of the oligodendrocytes are not going to be there to myelinate the axons. In the optic nerve, where that is highly myelinated, sometimes there is demyelination of the optic nerve that can result in blindness, double vision. There's also demyelination of the nerve structure that's associated with tracts that goes to the cerebellum, that is responsible for balancing, and sometimes the patient might have incoordination. There are a lot of areas where there is demyelination, And as a result, the patient might have specific symptom based on the area that is affected. So the ependymal cells also another cell. So we looked at the astrocytes, the oligodendrocytes. The ependymal cells are different type of cells. We already know that they line the ventricles and they help to produce the cerebrospinal fluid. They are similar to epithelial cells. They are cuboidal to columnar. They're lined in the ventricle and also in the central canal. So they're in the ventricle and the central canal. And these ependymal cells, it makes sense. These cells produce cerebrospinal fluid, and the cerebrospinal fluid flows around the ventricle, through the ventricle, lateral ventricle, third ventricle, through the aqueduct of Sylvius, down into the central canal, around the spinal cord or within it. So this is just showing the ependymal cells. I mentioned before that they have cilia or microvilli, apical projection. They also happen to have tight junction between them. Those are the only two important points about ependymal cell. They have cilia or apical projection, tight junction, just like the astrocyte that can form tight junction between the endothelial cells. And this is showing apical intracellular junction between the lateral borders, and you produce about 500 mils of cerebrospinal fluid daily. And these are produced by these flat cells that line the ventricle. This is just showing the choroid plexus. The choroid plexus is in the ventricle, and this is basically the area where the cerebrospinal fluid is produced. With the ependymal cell, we already mentioned that glial cells have the tendency to cause tumors, not the neuronal cells. But even the ependymal cells can cause tumor or glial tumors called ependymoma. This is rare. It's not as common as the other type of tumors. And naturally, because they're coming from the ependymal cells, you expect them to arrive from the ventricles of the brain, can arise also from the ventricle, from the central canal in the spinal cord. The microglial cells, we already said that those are the phagocytic cells that are there to fight against bacteria, viruses, parasites, whatever debris that gets into the central nervous system. And this is just showing the different layers on histology of the cerebral cortex. There is the gray, there is the outer gray area, the inner white area. And if you look at a histology slide, there are different layers, different category or classification based on the layers. So for example, you have the molecular layer based on the appearance, the molecular layer, granular layer, pyramidal layer, and external inner ganglionic multiform. And it's basically just describing the area based on what is in it. The molecular layer expect axons and dendrites. In the inner pyramidal layer, expect different types of cells, pyramidal cells. They have different names for these cells. Important cells that you guys should know are the neuronal cells, the glial cells, and which one you find in the central nervous system, which one you find in the peripheral nervous system, what are their functions. So those are the important things. In terms of the clinical correlation, there can be swelling of the cerebral cortex or the cerebrum, and as a result, you have cerebral edema. The cerebellum itself also has a molecular layer, granular layer, and you'll find Purkinje cells or Purkinje cell layer at different areas. You don't have to know the 7 molecular layers of the cerebrum or the 3 layers of the cerebellum. It's more understanding the cells. Yeah.
You went over the function of the satellite cells?
Not yet.
Okay,
so in terms of the spinal cord, remember that we said that the gray matter and the white matter is opposite. So for example, the brain, the gray matter would be peripherally located in the spinal cord. The white matter is peripherally located. So in the spinal cord, it's a bit opposite. And you know, it's based on where the tracks of the neurons are running and the cell bodies that are in that area. Yeah, this is just a repetition to reintroduce the spinal cord anatomy. You already covered this in the first few PowerPoints about the spinal nerves and the cervical, thoracic, lumbar, sacral, coccygeal. They're just different areas of the spinal cord based on what fibers or tracts of the axons that run running through different areas. For example, lateral horn, the anterior horn. The anterior horn, we already mentioned that you would have the motor axons going to neuromuscular junction or for motor function that would be in this anterior region. So there are just specific areas with tracts or nerve fibers for different function that will eventually end up in different areas in the body. It's just basically showing the cerebellar cell layers again, where you have the molecular layer, granular layer, as well as the Purkinje cell layer. This is just showing the Purkinje cell, giving a little bit more description of the Purkinje cells in terms of their shape and the amount of dendrites or axon that are projecting from them. And just like the brain, we expect that the spinal cord should have the dura pia arachnoid or dura arachnoid pia in the right order. Dura arachnoid pia, and we already know they're just connective tissue. So it's just the outermost to the innermost layer or region before it basically touches the spinal cord itself or the nerve fibers that are running through the spinal cord or along with the spinal cord. So there are peripheral nerves that extend from the spinal cord, and these will contact the muscles. These will contact different areas, different cells in the body. And just like the muscle that we looked at, the outermost layer will be the epi, just like we looked at the epimysium, endomysium. The outermost layer of the peripheral nerve will be epineurium instead of mysium. You'll also have the similar arrangement where you have the fascicles. Then there's the perineurium, however, that will surround the fascicle. What's special about the peripheral nerves? They're held together by tight junctions like the perineural cells, but we don't expect that the covering, like the perineurium, we expect them to be connective tissues as well. Loose connective tissue or dense connective tissue, depending on whether it's external or whether it's surrounding the nerve, the peripheral nerve itself, or whether it's surrounding the fascicle or the nerve fibers. You can try to understand this in your spare time if you want to. This is just showing, I mentioned it vaguely without the diagram, that there are different areas where the neurons are emerging from the spinal cord. These are either going to skeletal muscles, they're either going to smooth muscle, different areas in the body to carry out an action potential, simulate action potential for response. So it's basically communication from the brain, spinal cord to the end organ or the muscle itself. We already mentioned that in the peripheral nervous system, you have the Schwann cell that myelinate a segment of a single axon instead of multiple axons, unlike the oligodendrocytes that can myelinate multiple segments of different axons in the central nervous system. And we already mentioned the nodes of Ranvier. The nodes of Ranvier are just gaps in between each myelination where you have ion channel that will help to reinitiate the action potential. And we're almost done. This is showing a bigger picture of the nerve tissue or the neuronal cells. When, for example, they get to the muscle, this is contacting a smooth muscle. So when they get to the muscle, you have the Schwann cell that is here myelinating this neuron. This is showing a gland, submandibular gland, which showing one cell again myelinating this neuron. And this is showing a sympathetic terminal ending. And you also should have a parasympathetic terminal ending. Because remember that this is the autonomic nervous system. One is coming from the sympathetic ganglion, where they would inhibit salivation, or this would stimulate the gland to salivate. So this is just showing that there are actually nerve terminals that are in contact directly with the muscle cells or the gland cell or the cells of the gland, acini or mucinous cell, mucus or serous cells, if, for example, depending on where it is, or the myocytes or muscle cell. This is just showing blood vessel. Yeah, there are sympathetic terminals here as well. And that's the end of the presentation. In terms of the satellite cell, Daniel, they asked me about satellite cell.
Yes.
I had it in it. I deleted a lot of stuff. I didn't want to overwhelm you guys with this lecture because it's a lot of information, a lot of detail, and I had to prioritize what to share. But what I thought was important was the fact that you understood the basic neurological structure, understand the basic things like the neurons and the glial cells, and they have different function. Understand that the neurons or the nerve cells are just highly organized. They're a bit more complicated and much larger than the average cells. And once you understand that basic thing, you just know that the fibers, for example, axons, the cell bodies sometimes are located in the brain. The cell body might be in the spinal cord. Depending on where it's located, it might be a gray area. And extending from that cell body are axons. These axons can extend all the way down through tracts in spinal cord and communicate with presynaptic terminal to postsynaptic terminal in order to generate an action potential. But the bigger picture is that they're just cells communicating with cells. And with the nervous tissues, these cells are communicating in a highly complex way, but it's far more organized. So, I tried to minimize this as much as I could because I really didn't want to go over 100 and something slides. And I don't think you guys need to focus so much on all the specific details about it. As long as you understand that all these elements exist, and if you decide to further whatever you want to do, you can choose neuroscience if you want to. If you want to delve into neuroscience, it's a pretty interesting field, and there are a lot of detail where neuroanatomy is concerned. But I don't think I needed to mention all the nitty-gritty details for nerve tissue. Well, the satellite cells are there to are regenerative cells as well for myelination. The satellite cells, as well as the Schwann cell, can actually help in regeneration. For example, when you have nerve injury. I never read much more about the satellite cell because it's not really so commonly discussed.
Thank you, Miss.
Any questions?
Oh, Miss, I also saw the histology that you were showing with the different layers. Like that really requires a trained eye, doesn't it? Because it looks, all of them look the same to me, to be honest.
Yeah, and I understand, which is why I was like, how I learned histology was I had to go to the lab each time, look at it. I had to open my histology book, look at it, try to find pictures, try to, Google stuff, which is why I'll actually leave you guys with the microscope and slides to familiarize yourself with it, because that's the only way you will really understand it. You have to keep looking through the microscope, looking. Eventually, with the knowledge, putting the information from your head to the slides, eventually you'll understand it a bit more. But for biomedical engineering, you guys are cheated a bit of like a lot of things in terms of biomedical engineers. All of these things require exposure. In med school, you have to be at the hospital, like working alongside the doctors from third year, simply because it's exposure. You need exposure. I had to be in the lab in Vienna, I had to be in the lab here, just working. Even with the molecular stuff that I did, if I tell you how many times I troubleshoot and I got it wrong so many times, you wouldn't believe me. I spent a lot of time just leaving very late, until so much that I got it right eventually, and somebody's an expert in something that they weren't an expert in, because somebody was willing to take the time to be exposed to it repeatedly. The disadvantage for you guys, you just don't have most of what you need, but it's a work in progress. So hopefully things will change. And by the way, Daniel, the slides are not even good. They're not like the best image, and we don't have the best microscope either. So you're really struggling with less. But it's also good because if you can learn from what you have that is not the most optimal, then you'll do far better when you have everything, or you can even learn how to improvise. So I don't see it as a big issue. Primary thing is to get you guys exposed to a lot more of the laboratory component of it, where you can add your relevance to the field. So if you want to branch off into electrical engineering, if you want to do the biomedical engineering or the advanced molecular engineering, go into instrumentation, for example, biomedical instrumentation. If you want to go into app creation or some technology related part of engineering which exists, then you at least need the exposure. Yeah, so that's that. Any questions or concerns? Any desires that you guys would have for this course that will help you that I probably have not explored?
I can think of right now.
So I think that's it.
Thank you, Dr. Lee.
No problem.
Thank you, Miss.
All right. Bye.
Bye.